So after radiation chemistry, biochemistry, and radiation physics, chemistry, and biochemistry, we move now again one step ahead and we reach the cellular level. And in particular, we will look today at the cell killing induced by radiation. Cell killing, which is, as we will see, one of the most important endpoints. Certainly the most important for radiation therapy, but also important for radiation protection, at least when you talk about high doses, where actually you can have cell killing in that case. So at the beginning, we have to discuss a little bit, this will be the first part of the lecture, we'll focus on what is cell death. So we are not talking about an organism now, we are still at the cellular level. So what does it mean, cell death? Generally speaking, a cell is dead if is unable to replicate or is unable to function normally. There are also cells like neurons that do not replicate at all. In most cases, when you talk about cells in culture by definition tends to grow. So the cell killing means stopping their replicative ability. As you see, there are at least 5 pathways of cell death, and these are listed here in this review. So apoptosis is the first one. The characteristic are shrinks, a condensed chromatin, blebbing of cell membrane. Here are the methods that you have to use if you want to measure this kind of cell death. Typically, apoptosis is a programmed cell death. So it's the cell that decides to go into apoptosis. Vice versa, necrosis typically is not genetically determined. So it's induced externally. If you burn your skin with the cells, with a cigarette, you induce necrosis in the cells. You have a breakdown of the cell membrane, atypical nuclear shape, vacuolization, disintegrated cellular organelles, and so on. Mitotic catastrophe is something that occurs during mitosis. So this is a very important mechanism in radiation-induced cell death. Of course, you can also have apoptosis. You can have all of them after radiation, but mitotic catastrophe is important because it's strictly related to the chromosomal aberration. So you see, it's caused by missegregation of chromosomes and dorsal fusion. Cell often a micronuclei, which is, we didn't touch them in the chromosomal aberration, but it's also an expression of the loss of genetic material in the cytoplasm. And you can see giant cell formation. Senescence, which is also important, very important in radiotherapy, that's when essentially the cells are still metabolically active, but non-dividing, and show an increase in the cell size. So essentially they are not really completely dead, but they don't grow anymore. That's what can happen to tumor cells. Tumor cells sometimes go in senescence, and this is good, of course, because then they stop growing. Because the cell express a beta-galactosidase enzyme, it's easy to stain them for senescence. With beta-galactosidase, they give you immediately an idea whether the cells are going in senescence or not. And finally, autophagy. Autophagy is also a form of programmed cell death, like apoptosis, but is characterized by the formation of double membrane vacuoles in the cytoplasm, which sequester organelles such as mitochondria and ribosomes. So it's a sort of like is wallowing itself, creating membranes, say, to kill itself. And while apoptosis is depending on p53 and caspase, p53, of course, we have seen why, autophagy is independent of p53 and caspase. So we will now see some more details on this cell death pathway. Now, you see here again a little bit the different types of cell death that you can have. You see here the mitotic in this growth. I mean, from the mitosis, and the different outcomes that can be cell death, growth arrest, mitotic death, which is essentially a form of mitotic catastrophe. Mitotic death here, if you have chromosomal aberrations, senescence, aneuploid cells, and so on and so on and so forth. So this is what happens when you have abnormal cell division, cytokinesis failure, or mitotic slippage. So it is, then you see, for example, if you have mitotic slippage here, you can have octoploid cells, aneuploid cells. And this is what happens in mitosis. We will go back to the mitosis. So let's start instead with indeed with mitotic with mitotic catastrophe, because you see the role, the chromosomal aberrations when you go into mitosis eventually will result in abnormal cell division, and this can lead directly to mitotic death, to mitotic catastrophe. You can see it nicely here. You see, these are cells, these are HeLa cells. HeLa cells are very well known tumor cells that comes from this lady that died of breast cancer many years ago. These are fluorescent images expressing the histone 2B in cherry, so in red, and the alpha tubulin in green, because this is EGFP, the green fluorescent, expressing green fluorescence protein. So this is in live cell microscopy after treatment with DNA damage. It's not really radiation here, but will be the same with radiation. So you see in this movie, you can see a normal mitosis. So these are now the flesh. You see, essentially, you see H2B is staining the nucleus of the cell, and you see it in red. And the alpha tubulin, which is instead the cytoskeleton in the cytoplasm, is clear in red. Now, at a certain point, you are seeing it here. You see nicely the cell division, the mitosis in these cells, very nice mitosis, and is now split into two cells, again, with two nuclei and two cytoplasms. But here, you will see instead that when these cells tries to go into mitosis, instead, it has a mitotic catastrophe. You see, it's irregular. The splitting of the nuclei is inevitably irregular, and the cell goes in mitotic death. So the mitotic catastrophe is a consequence of chromosomal aberrations, as you see. Cellular senescence instead has a different origin. You see here the staining with beta-galactosidase. These are cells, normal cells in vitro. These are primary mouse embryonic fibroblasts. You see their shape is kind of spindle, is long, like fibroblasts should be. Fibroblasts are the cells of the connective tissue. When they become senescent, they become more flat, and they are now positive to the stain in beta-galactosidase. So the senescence can occur spontaneously. It's the Hayflick limit that we have seen about in the biology lecture when we mentioned telomeres. Now, though they no longer replicate, senescent cells remain metabolically active and generally adopt phenotypes including flattened cell morphology, you see here, altered gene expression and secretion profiles. These are senescence-associated secretory phenotypes, so you also detect some molecules called cytokines in the medium that are associated to senescence. As I say, this is a mechanism, it's a very good cell death mechanism for tumors, because if tumors go in senescence, they will not grow anymore. Autophagy, this is relatively recent and still under study, is a catabolic mechanism that involves cell degradation of unnecessary or dysfunctional cellular mechanisms through the action of lysosomes. So in general, generally speaking, is a mechanism, you see it here, when you can induce autophagy in the cells. You see, chemotherapy, targeted therapy can induce autophagy. Typically, if you treat cancer cell with chloroquine, chloroquine that is now becoming a very, very famous molecule also for the COVID-19 as a possible in COVID-19 disease. This leads to deacidification of lysosome followed by an accumulation of ineffective autophagic vesicles, you see it here. In cells depending on autophagy for survival, autophagy inhibition with chloroquine leads to cell death. So somehow autophagy can be, you can die if you inhibit autophagy, or vice versa, autophagy can induce cell death. You see, these are cells, prostate cells treated with chloroquine. You see, in this specific case, the autophagy is able to isolate dysfunctional cellular components, and so this cell will survive. But if you inhibit autophagy, then these vesicles accumulate and eventually the cell dies. So it's a very complex mechanism. It's quite new. I mean, it was not very well known in the past, but it's taking now more and more importance in the explaining cell death in tumors. And he said apoptosis is now pretty. This is a comparison between necrosis and apoptosis. Necrosis, as I said, is a passive mechanism of cell death. Essentially, for example, if you have exhaustion of oxygen or nutriment, exhaustion of ATP, impairment of the cell membrane, and then you will have enzyme release that cause inflammation. Apoptosis instead, you see it here, you see viable cells that goes in this direction goes into apoptosis, and in this direction goes into necrosis. And you see here some examples, you see, of cells going into apoptosis or necrosis. So in apoptosis, the mechanism is not so easy. You see, it is simplified here. The caspase-3 plays a very important role in apoptosis. There must be a trigger of the death program, generally decided by the p53. And then from the p53, you have an intracellular signaling which is mediated by the caspases, by the protein caspases. You start to have fragmentation of the cell into vesicles, and eventually you will have phagocytosis of the apoptotic body. Instead, you see in necrosis, you have swelling, blebbing, and release of proteolytic enzymes. So essentially, necrosis will be a passive death, and apoptosis will be an active death. In radiation biology, necrosis is actually seen when you have very high doses. You can have necrosis also in the brain, for example, when you apply very high doses for radiotherapy, and this is a toxicity of radiation, something that you don't want. Apoptosis, unfortunately, you don't see it very often in tumors because generally tumors are either mutated in p53 or they have further mutation in caspase, so that they can escape apoptosis. In fact, one of the strategies to treat the tumors is to reactivate apoptosis in the tumor so that they can actually go into apoptosis. In fact, I mean, apoptosis is quite a common process. It's not so uncommon. This is now a little bit more the detail, let's say, of the apoptotic process. It's remarkable that we have approximately 10 to the 6 per 10 to the 10 cells that die every day in our body by apoptosis. And this can be by extrinsic factors like hormones, radiation, or intrinsic inducers. The pathways go through mitochondria, signal transduction, and calcium concentration. As I said, common regulators are p53 and caspases. Now, if you have excess apoptosis, you go into atrophy. Into atrophy. This is known. I mean, the the atrophy disease are very often associated to excess apoptosis. You also remember the mouse that was expressing too much p53 goes into accelerated aging and had an atrophic phenotype, which corresponds to excess apoptosis caused by p53. Vice versa, of course, if you inhibit p53, this can lead to cancer, and this is in fact a strategy. It's very commonly found in cancer. At least 50% are mutated in p53 and overexpress BCL2, which is a protein downstream of p53. It has to be repressed to go into apoptosis. If it is overexpressed, you will prevent apoptosis. Now, only tumors originated from hematopoietic and reproductive tissue are sensitive to apoptosis. Those tumors, like the liquid tumors, like leukemias, lymphomas, or those in the reproductive tissues, for example, a testis tumor or ovary tumors, those cells go into apoptosis because these cells in the hematopoietic reproductive tissues are naturally use apoptosis a lot to regulate their number. So it's not surprising that those tumors are still sensitive to apoptosis, and in fact, in generally, they are radiosensitive tumors. It's relatively easy to kill them because they are radiosensitive, because they go into apoptosis. Now, this cartoon is not to scare you, of course, but it's just to show you the complexity of the problem. I mean, we are all physicists in this course, and the biologists always think that physics is difficult, it's complicated. It is not. I always say this is complicated. You see, this is really complicated. This is the pathway where you see the involvement of the many different. This is the membrane, actually. This is the cytoplasm where you see this caspase-dependent cell death, you see. These are the mitochondria that also play a key role. And finally, here is the DNA. You see, for example, you can start to read this cartoon. You can start from radiation here, DNA damage. You see the ATM, for example, which, as you know, is one of the key player in dealing with radiation-induced DNA double-stranded breaks. This we have seen before. ATM phosphorylate the CHK2, a checkpoint protein, which in turn activate p53. And now, through the activation of p53, you can go, for example, via this pathway that acts on the mitochondria, on bugs, you see, and eventually brings you to a caspase-independent cell death. Or you can take other pathways. For example, you can, if you inhibit survivin, you will have another pathway, and so on and so forth. So the cell death pathways, as you can see here, are really amazingly complex, and the number of proteins involved are also a huge number. You will understand that if you want to have a pharmacological intervention, you have to understand all this protein, and you have to understand the role of these proteins, because then eventually you have to target them with a specific drug. The story is sometimes even more complicated. I told you that necrosis is actually a non-programmed cell death, is induced by external agent, but is not programmed. But this is not always the case. You see here that there is a pathway, for example, pathway which is dependent on RIPK1. This kind of pathway, again, if you have a ligand here on the cell, or if you have the impact of some kind of damage, you can go into these pathways. One of them will bring to survival, depending on the regulation of FLIP. You see here how the transcriptional regulation plays a key role. If you have high levels of FLIP, you will have eventually survival. If you have no FLIP, you will have eventually apoptosis. And if instead you inhibit caspase-8, which is, you see here, is a key part in in the apoptotic process. The cell goes into necrosis now, but now this is not a natural necrosis, it's a programmed necrosis. So sometimes you see there are even cases where you can have necrosis not because of the high doses and the external agent, but as a programmed pattern when you have, for example, inhibited caspase-8. In some tumors, this can happen. So now this is, of course, the most difficult part, I think, in this lecture, because it's the part of molecular biology, but it is necessary that you will remember, let's say, that there are different pathways for that, at least 5 we have seen. Some of them are programmed cell death, some of them are non-programmed cell death. Radiation can induce all 5 of them. For sure, mitotic death is a very important pattern after radiation because of the induction of chromosomal aberrations that we have seen, but all the others are also possible. Now, how do we measure cell killing in radiation studies? So essentially, you see, let's start from the simple case of when we grow the cells in vitro. So we are not working on animals, we are not working on patients now. We are working on cells that grow either in petri dish, like here, or in bottles, like here. This red liquid that you see on top is the growth medium. It's something you have to give to the cells so that they can grow. Under these conditions, I think I showed you already this slide. Under this condition, the cell grow. The grow kinetics has a lag phase initially, followed by exponential growth, which is regulated, of course, by this equation. You know, it's essentially every cell duplicate. So it's 2 to the population doubling number, but the population doubling number can be written as T divided by TD, where TD is the duplication time, the average duplication time. And eventually, if you like exponential, as I do, then you prefer to write it as N0 e to T divided by tau. And of course, tau is TD divided by the logarithm of 2. And then eventually you will plateau here, or even you go in cell death if you don't have enough medium now, the cells cannot grow anymore. Medium is not enough. They will go in necrosis or they will go in senescence. They start to die, actually. So you can grow the cells and you can irradiate them either when they are growing or also when they are in plateau, when they are stopped. When they are in plateau, they go in G0 phase. G0 phase is actually the most common phase in our body. Most of the cells in our body are in G0 phase. They are not growing. There are only the cells, mostly cells in hematopoietic tissue that we saw already. They divide continuously. We produce billions and billions of new hematopoietic cells every day. And those in the intestine too, they also proliferate quite quickly, but most of the others stay in G0. So irradiating the cells in G0 is also is practical because it represents a lot of normal tissue. Irradiating them in exponential phase, though, give you an idea of the different sensitivity of the different phases of the cell cycle. So how do we measure the cell killing? Of course, you can say, okay, we can use beta-galactosidase to measure senescence, whatever. But at the end, when we have cells that are growing, we want to see whether they are able to grow. So the definition of cell killing in radiation biology is generally clonogenic cell survival. So the ability of a cell to generate a clone. You see that you start from day 1 here. This is day 2, day 3, day 4, day 5, day 6. The rule of the thumb says that after approximately 7 days, because you know the TD in human cells and in most mammalian cells, but human cells in particular, is approximately 24 hours. So the rule of the thumb says that if you have a colony with more than 50 cells after 7 days, this is a survivor. So if you start with 1 cell, if you plate, let's say, 1,000 single cells after irradiation, you take the cells, you plate them after irradiation, you plate them 1000, if after 1 week you have 500 colonies larger than 50 cells, this gives you a survival of the 50%. That's the simple definition. And in fact, experimentally, this can be done in this way. You see, you take your cells from a bottle, for example, you trypsinize. Trypsin is an enzyme that removes the cells from the surface, so they go in suspension. And then you can plate them. For example, you can plate them at different densities. Why at different densities? Because you want to apply different doses. And then here, low dose, you want to put only a few cells. But at high dose, you want to put a lot of cells, because otherwise you will not find colonies, because you kill them all. In fact, then you irradiate, you incubate, and then you count the colonies. You see, for example, here, you have 100 cells, and then you count 9 colonies. So this means that here the survival is actually 9%. But this is not called survival. This is called plating efficiency. So it's because of course these cells were not irradiated, but even though they were not irradiated, not all the cells are able to form colonies. So there is a fraction of the cells that are unable to form colonies. You have to normalize this number to have the surviving fraction. You see here, for example, you plated 400, you find 8 colonies. So the plating efficiency is 0.02, but the survival now is 0.22. And 0.22 comes from 0.02 divided by 0.09. So to have the survival, you always must have a control, and your control gives you the plating efficiency. When you calculate the survival, you have to know how many cells you plated, you have to count the colony, and eventually you have to divide by the plating efficiency. And this is essentially written here. At the end, generally, what happens when you do this work, so you see, you count these colonies. This is generally done really manually at the microscope with the low magnification, because these colonies are really big. You see, this is actually essentially is slightly smaller than the real size, but I mean, they are very visible, easy to study. There are many cells. These days, there are also automatic scanners, that can count the colonies automatically, but in the old time was done manually. It still is done very often manually because it's actually very easy. And then you build the curve. You calculate the survival in this way, like number of colonies counted, divided by the number of cells that you had plated, also counted through a cell counter, though, not, of course, not one by one. You count through an automatic cell counter. And you have to normalize with the plating efficiency. Very important, because the plating efficiency is generally not 1. Generally, if you plot these numbers for many doses of X-rays, you end up with the linear quadratic curve. But be careful, it's linear quadratic in log scale, so it's an exponential of minus alpha d plus beta d squared. So we saw already, I told you already when we saw the microdosimetry that everything in biology is linear quadratic. It is linear quadratic also here, but as you understand immediately, the survival will be e to minus alpha d plus beta d squared, because if the lethal lesions are equal to alpha d plus beta d squared, then the probability of having 0 lethal lesions, according to the Poisson statistics, will be e to minus alpha d plus beta d squared. So this is a linear quadratic, typical linear quadratic. The exponential is only because you apply a Poisson statistics, and the survivors are those cells that will have 0 lethal lesions. And the probability of having 0 lethal lesions is e to minus alpha d plus beta d squared if the number of lethal lesions per cells is equal to alpha d plus beta d squared, as we have seen already previously in microdosimetry. And as we have also seen that chromosomal aberrations are linear quadratic. Double-stranded breaks, if you remember, are not linear quadratic. They are strictly linear. But chromosomal aberrations are instead linear quadratic because they can be induced intertrack or intratrack. The ratio alpha over beta the dimension of this ratio is gray is extremely important, because this is the contribution from the quadratic term, let's say, let's put it this way. So a very high alpha beta ratio means that this curve is almost straight. A very low alpha beta ratio, this means that this bending in the curve is very significant. So the alpha beta ratio gives you an idea of the radiosensitivity of the cells, because we will see beta is related to the repair. So very radiosensitive cells have very high alpha beta ratio, very radioresistant cells have very low alpha beta ratio. So why alpha beta, let's say, why linear quadratic? This essentially we already saw when we treated chromosomal aberrations, but let's see a little bit more. Essentially, in the microdosimetric approach, and we will see that there are many different models, but in a microdosimetric approach, you imagine that you have a linear term which is due to single electrons or single alpha particles, I mean single radiation tracks that induce two different lesions. So this is an intra-track event. This mechanism, we have two chromosomes here. So we are talking about microns. We have two chromosomes here. This mechanism is proportional to the dose. But you can have two lesions leading to a dicentric, for example, also if you have two independent electrons. These terms is proportional to dose times the dose, so it's proportional to the quadratic terms. So the number of lethal lesions, so the idea is always that you must have at least two lesions to have a cell killing. We will see, this is, of course, this is true, approximately true for mammalian cells, because, you know, you see, you need the chromosomal aberration, and the chromosomal aberrations are always required at least two breaks. But if you take viruses, for example, this is not true anymore, but for chromosomal aberration. But this is not a problem, because if you put beta to 0, this becomes linear, of course. So eventually, this mechanism, you see, of intra-track plus inter-track mode gives you a linear quadratic induction of lethal events, and the survival will be 0 lethal events, Poisson statistics, E2 minus the average, which is alpha d plus beta d squared. Of course, as I told you, the alpha beta ratio characterizes very easily the radiosensitivity of the cells. You see, repair proficient cells that tend to have a low alpha beta ratio, a very pronounced shoulder, you see here, and instead repair deficient cells down here, where the alpha beta ratio is so high that essentially beta is 0. So you see that here you have also different tumor cells that have different sensitivity in vitro. We will see that the sensitivity in vitro is not necessarily the same as the sensitivity in vivo, but already in vitro, let's say, you see that they are fairly different radiosensitivity of different tumors, and within the same tumor, these are all lung cell lines, you see. Also the same within the lung, you have more sensitive and more resistant cell lines. The target for cell inactivation, we have discussed this many times, is certainly the DNA. One of the main evidence from this is very old, actually, an experiment done in the 60s or early in the 70s, you see, from Munro, 1972. He was using a needle of polonium, and he was, because polonium is alpha emitter and alpha particles have such a short range, he could put the needle in the cytoplasm or in the nucleus, and he clearly showed that if he put it in the cytoplasm, there was survival. If you put it in the nucleus, the cell will die. So essentially, this is a very old experiment, but makes the point that we have done already many times, so that the nucleus is the main target. These days, you will see this is not necessarily true. So you can have cell death by cell-to-cell communication, by signaling transduction in the cytoplasm, but let's say that is by far the main target. The other are minor effects. They can be important in some cases. Also, classically, of course, now with our molecular biology knowledge, we have to try to transform these words in a more modern way. But classically, you call lethal damage irreparable severe damage with resulting cell death. This can be, you understand, this can be a severe clustered lesion, a complex clustered lesion that cannot be repaired and bring the cells into apoptosis, but can also be, for example, a misrepair event. I mean, something that come to the misrepair events, like a dicentric. However, the misrepair event comes from sublethal damage. So you can say that one single double-stranded break is still sublethal, but if it interacts with another sublethal double-stranded break, then you will have finally a lethal event. And this is why you have this alpha D plus beta D squared, this intra-tract and inter-tract induction of sublethal events. And then you have potentially lethal damage. So even if you have lethal damage, this can be potentially lethal, because this can be, depends on the time of repair. So if you wait enough time for repair, then you will have more survival. This is so in, for example, if you irradiate cells in plateau and you replate them for cell counting only after 1 day, you will see higher survival than if you plate them immediately. So some damage is potentially lethal, but can still be fixed if you leave the cell to repair them. And this is, if you remember, the rejoining curve of DNA double-strand breaks as a fast component, but also as a slow component, and this slow component is up to 24 hours or more. So it's not so strange. Now, the correlation, let's say, between radiation and cell death is of course, is very complex. We know a few things. We know that single-strand breaks, generally speaking, are not lethal. We know that double-strand breaks also generally are not lethal, can be repaired. We have seen that there are sophisticated mechanisms of repairing double-strand breaks, but complex clustered double-strand breaks, those becomes more lethal. There are some people that say that the initial double-strand breaks correlate with lethal events, but it's more likely that the residual double-strand breaks correlate with lethal events. Depends on what is a double-strand break. So of course, if you take complex clustered, then it's more likely that you go into unrepaired breaks apoptosis, or that you go, for example, in alternative non-homologous end joining, and then you have a translocation, or a dicentric, or a asymmetrical interchange, acentric fragments that will lead you to mitotic catastrophe. In fact, you see chromosomes are bidirectional involved in cell killing, and in general, you clearly see there are many different mechanisms for cell killing. So having a simple explanation, a simple model is not so easy. We will see how the models, we will see now, how the models evolved in the history, and how they have to become more and more complex if they want to take into account now the huge molecular biology that we know. I will only describe you simple cell killing model here. I will not go into the detail of the modern models. They try to start from the DNA repair damage. So they try to model the kinetics of the enzymes of the repair, so that you know that they start really from ab initio and then they go on. But these models are so complex and the uncertainties is so high because we don't know. You have seen, I mean, that we don't really know exactly what the proteins are doing, that I will leave them away from now, and I will only talk you about some, let's say, simpler but somehow more practical models of radiation action. And this I will do in the next lecture.